Welcome friends to a new session on genomics and proteomics. In this session, we will discuss biological techniques in genomics. According to the central dogma of molecular biology as enunciated in 1958 by Francis Crick, DNA were transcribed into RNA to the basic operational elements of cellular biology, proteins, whose interactions through many levels of complexity result in the functioning of living cells. This process is called gene expression. This was first description of the action of genes. Remarkable technologies such as microarrays and their descendants, high throughput sequencing, in vivo imaging techniques and many others have enabled the biologists to begin to analyze the function of gene at molecular level. The various aspects of these analysis have coalesced as omics, transcriptomics, the study of gene to gene regulation, in particular DNA protein interactions, proteomics, the large scale study of the structures and functions of proteins, metabolomics, the study of small molecule metabolite profiles. Genomics, which encompasses all these, can be thought of as a science linking DNA structures with functions at the cellular level. Technologies like the polymerase chain reaction (PCR) as we call, automated DNA sequencing and microarrays for sequencing DNA, RNA and proteins have made possible the direct study of genetic material, and molecular biology entered the genomic era. Let us now look into the basic tools of gene exploration. Basic methods have been combined in groups reminiscent of the complexity of genomes to generate further higher levels of data. The results of thousands of assays have been collected in databases such as the NCBI and the Ensemble genome browser. These databases are enabling us to begin to trace the steps from genome to regulome to proteome to metabolome and the fundamental interactions of genotype and the environment that lead to the observable phenotypes and diseases. The rapid progress in biotechnology is a result of few basic methods for gene exploration: DNA cleavage by restriction enzymes and separation by gel electrophoresis, blotting techniques, DNA sequencing, solid phase synthesis of nucleic acids and the polymerase chain reaction. Let us now discuss restriction enzyme analysis. The term restriction enzyme originated from the phage lambda and the phenomenon of host controlled restriction and modification of a bacterial virus. The phenomenon was first identified by Salvador Edward Luria and Giuseppe Bertani in the early 1950s. It was found that for a bacteriophage lambda that can grow well in one strain of Escherichia coli, for example E. coli C, when grown in another strain, for example E. coli K, its yields can drop significantly. The host cell E. coli K is known as the restricting host and appears to have the ability to reduce the biological activity of the phage lambda. If a phage becomes established in one strain, the ability of that phage to grow also becomes restricted in other strains. In the 1960s, it was shown in work done in the laboratories of Werner Arber, who shared the Nobel Prize, and Matthew Meselson, that the restriction is caused by an enzymatic cleavage of the phage DNA, and the enzyme involved was therefore termed as the restriction enzyme. The restriction enzymes studied by Arber and Meselson were type 1 restriction enzymes, which cleave DNA randomly away from the recognition sites. In 1970, Hamilton Smith, Thomas Kelly and Kent Wilcox isolated and characterized the first type 2 restriction enzyme HINE2 from the bacterium Haemophilus influenzae. These types of restriction enzymes are more useful for laboratory work because they cleave DNA at the site of the recognition sequences. Later, Daniel Nathans and Kathleen Dana showed that the cleavage of simian virus 40 SV40 as we call them DNA by restriction enzymes yields specific fragments that can be separated using polyacrylamide gel electrophoresis (PAGE), thus showing that restriction enzymes can also be used for mapping DNA. Werner Arber, Daniel Nathans and Hamilton O. Smith were awarded the 1978 Nobel Prize for Physiology or Medicine for the discovery and characterization of these restriction enzymes. Restriction enzymes or restriction endonucleases are able to scan along a length of DNA looking for a particular sequence of bases that they recognize. This recognition site or sequence generally varies from 4 to 6 base pairs in length, have been discovered in many organisms, especially bacteria and single celled organisms. It recognizes specific base sequences in double stranded DNA and cleaves at specific sites and both the strands containing recognized sequences. The size of these fragments is measured in base pairs or kilobase pairs. This is very helpful and indispensable for analyzing the chromosome structure, gene isolation, sequencing of long DNA molecules and creating new DNA molecules that can be cloned. Restriction endonucleases are found in a number of prokaryotes. Their biological role is to cleave foreign DNA molecules. The prokaryotic cell's own DNA is not degraded because the sites recognized by its own restriction endonucleases are methylated. Many restriction endonucleases recognize specific sequences of 4 to 8 base pairs and hydrolyze a phosphodiester bond in each strand in this region. A striking character of this restriction site is that they always possess two-fold rotational symmetry, or the recognized sequence is a palindromic or an inverted repeat. Restriction enzymes cut the DNA at specific sites and these sites are determined by the sequence of the bases, which usually form palindromes. Palindromes are actually groups of letters that read the same in both the forward and the backward orientation. In the case of DNA, the letters are found on both the forward and the reverse strands of the DNA. For example, the 5' to 3' strand may have the sequence GAA TTC. The complementary bases on the opposite strand will be CTT AAG in the 3' to 5' direction, which is the same as reading the first strand backwards. Many enzymes recognize these types of sequences and will attach to the DNA at this site and then cut the strand between two of the bases. The sequence of the restriction enzymes EcoR1, HIND3 and BAMH1 are shown in the figures 1 and 2. The cut sites are indicated by arrows. Figure 2 shows the size of each of the fragments or bands that were produced when lambda DNA was cut with each of these restriction enzymes. The sizes were determined by comparison to a molecular ladder or marker which has bands of known sizes when it is separated by electrophoresis at the same time as the digested lambda DNA. Specific DNA repair enzymes will re-anneal or stick these DNA fragments themselves to other fragments with cut ends that are complementary to their own end sequence from the same organism or even from a different one. This ability of DNA to repair itself has been utilized by scientists to introduce foreign DNA into an organism. This DNA may contain genes that allow the organism to exhibit a new function or process. This would include transferring genes that will result in a change in the nutritional quality of a crop or perhaps allow a plant to grow in a region that is colder than its usual preferred area. The discovery of restriction enzymes allows DNA to be manipulated, leading to the development of recombinant DNA technology that has many applications, for example, allowing the large scale production of proteins such as human insulin used by diabetics. Restriction fragments can be separated by gel electrophoresis. Gel electrophoresis is a laboratory method used to separate macromolecules, which are mixtures of DNA, RNA or proteins, and their fragments according to the molecular size. In gel electrophoresis, the molecules are separated or pushed by an electrical field through a gel that contains small pores. The molecules travel through the pores in the gel at a speed that is inversely proportional to their lengths. This means that a small DNA molecule will travel a greater distance through the gel than larger DNA molecules. In gel electrophoresis, the gel refers to the matrix used to contain and then separate the target molecules. It is a cross-linked polymer whose composition and porosity is chosen based on the specific weight and composition of the target to be analyzed. When separating proteins or small nucleic acids, like DNA, RNA or oligonucleotides, the gel is usually composed of different concentrations of acrylamide and a cross-linker, producing different sized mesh networks of polyacrylamide. Acrylamide, in contrast to polyacrylamide, is a neurotoxin and must be handled using appropriate safety precautions to avoid poisoning. Agarose is composed of long unbranched chains of uncharged carbohydrate without crosslinks, resulting in a gel with large pores allowing for the separation of macromolecules and macromolecular complexes. Gel electrophoresis is a method for separating molecules according to their rate of traversal across an electrified gel. Agarose and polyacrylamide gels are the commonly used gels. Electrophoresis refers to the electromotive force EMF that is used to move the molecules through the gel matrix. By placing the molecules, the DNA samples, in wells in the gel and applying an electric field, one end of the gel has a positive charge and the other end has a negative charge. Because nucleic acids, DNA and RNA are negatively charged molecules, they will be pulled towards the positively charged end of the gel. Nucleic acids will move through the matrix at different rates determined largely by their mass or base pairs when the charge to mass ratio of all the species is uniform. Finally, after the DNA or RNA molecules have been separated using gel electrophoresis, bands representing molecules of different sizes can be detected as seen in the figure 3. The molecular weight size markers, which are mixtures of molecules of known sizes, run on one lane of the gel parallel to the unknown samples. The bands observed can be compared to those of the unknown in order to determine the size. There are a large number of buffers used for electrophoresis, which are used to provide ions that carry a current and to maintain a pH at a relatively constant value. The most commonly used nucleic acid tris acetate EDTA, tris borate EDTA, TAE has the lowest buffering capacity, but provides the best resolution for larger DNA, means a lower voltage and more time, but a better product. The DNA in the gel can be stained after electrophoresis and may be visualized using ethidium bromide, which when intercalated into the DNA fluoresce under ultraviolet light. Other methods may also be used to visualize the separation of the mixture's components on the gel. An autoradiograph of the gel can be recorded in case of radioactive molecules. The photographs can be taken of the gels using a gel documentation system.